Okay, so let's now move to the next presenter, that is James Dowsett. James is our third place winner. He's a long-time brain products user and is doing a postdoc at the Department of Neurology at LMU here in Munich, in Germany. His paper is published in a Journal of Neuroscience Methods, and the title of this work is Mobile Steady-State Evoked Potential Recording, Dissociable Neural Effects of Real-World Navigation and Visual Stimulation. The judges praised the high degree of mobility in this setup, and particularly liked the method for measuring and removing data noise. They also liked its design, as it allowed for interesting results with spatial attention. So if James is ready, I would like to give him the presenter rights, and we look forward to his talk.
Okay, so thank you for that introduction, and thank you for giving me the opportunity to present my research. So the method that I'm working on is mobile SSVEPs. So firstly, why are SSVEPs interesting? Well, what we're really interested in is neural oscillations. So we know that neurons oscillate, populations of neurons oscillate, and neural oscillations are really fundamental to how the brain works. So to understand the brain, we want to study them. And the way in which we can study any system which oscillates can be divided into two broad strategies. We can observe it and see what it does, or we can drive it. We can force it to oscillate and see how it reacts to inputs at different frequencies. And these are two equally valid strategies for understanding neural oscillations. And in EEG research, this broadly maps onto induced versus evoked paradigms. And there's many ways which you can drive neural oscillations by stimulating the senses at specific frequencies with flickering light, with sound, and with rhythmic tactile stimulation. And here I'm going to focus on the so-called steady-state visually evoked potential, the SSVEP. And these are rhythms in the brain which are caused when a visual stimulus flickers at a specific frequency, and we see the strongest effects when this frequency is close to the brain's natural oscillations. So just to illustrate this point, here is some data that I've recorded on myself. Here is my resting brain activity in blue, showing the familiar dominant alpha rhythm of about 10 Hertz. And in green are the SSVEPs. These are responses to visual flicker at different frequencies, at 5, 5.5, 6, 6.5, and so on. And if we track the amplitude of these responses, we can see that more or less they follow the peak of the brain's natural frequency. Now, just to be clear, when I'm talking about the SSVEP today, I'm talking about the average phase-locked response. So here we have the light, flickering light, flashes on and off, and with every flash of the flickering light, the EEG signal is segmented, and these segments are averaged together. So any signal which is time-locked or entrained to the flicker will contribute to the average, and anything that's not time-locked will eventually average to zero. Now this presents a solution to the problem of motion artifacts. If we average enough trials, any source of noise that's not phase-locked to the flicker will be lost in the process of averaging. If we average enough segments, we can eventually get rid of any source of noise, regardless of how large it is. It's simply a question of collecting enough segments so that it averages to zero. So how can we make these SSVEPs? Well, historically in the literature, there's generally two ways in which people do this, either with flashing LEDs, in this case on the left, directly into the eyes. And this has the advantage that it can be made to flicker at any frequency, but LEDs are not very interesting. It is just a flashing light. Now, the second way is to create SSVEPs with images on a screen flickering at different rates, and this is commonly used in brain-computer interfaces. Now, this is more interesting because with frequency tagging, you can have different elements on the screen flickering at different frequencies and isolate these frequencies in the EEG. So there's more you can do in terms of experimental design, but you do need to be looking at a screen. Now, although many of us spend a lot of time sitting in front of a screen nowadays, this is not what our visual system evolved to do. The real world is far more interesting and important for truly understanding how the brain functions in everyday life. And for mobile EEG, of course, we don't want people just to be sitting in front of a screen. We want people to moving around in the real world. So what I'm presenting today is a new method for producing SSVEPs using LCD glasses. Now, these glasses are typically used for 3D TV to rapidly block one eye at a time and create the illusion of 3D. The way it works is that the LCD glass darkens whenever a voltage is applied across it. So here I've adapted them to be controlled with a microcontroller so they can flicker at any frequency. And this method has a number of advantages. Firstly, evoked potentials can be generated from anything the participant is looking at in the real world. So this is not limited to flashed images on a screen, but this is rather a flickering view of the real world. Now, this is important because visual cognitive neuroscience is generally focused on pictures and simple stimuli presented on screens. But we cannot assume that the brain activity we get from pictures of visual scenes and pictures of places maps onto the brain activity that occurs when we're in real places and real spaces in which we can move around. Natural scenes will accurately represent the functioning of the brain in ordinary life. So although virtual reality is getting better all of the time, it's still a long way from recreating the full richness of the real world. And real life situations will inevitably involve emotional salience and real consequences to actions which are absent in virtual worlds. A second advantage is that unlike SSVEPs on a screen, which are limited to the multiples of the refresh rate, here we can create SSVEPs at any frequency. So for example, we can do a frequency sweep through the alpha range, like I showed just before, and also we can go up into the gamma range, and we can track how the signal resonates with, or entrains, the underlying neural circuits. And a further advantage is that recovering clean SSVEPs only requires one electrode. So eventually this method could be incorporated into a small discrete headset that could potentially be worn throughout a normal day. So although amazing things can be done with large numbers of electrode, such as source localization, no one's ever going to really walk down to the shops with 128 electrodes attached to them. So this is something that could potentially be worn in the real world. Although a combination with large numbers of electrodes for source localization of the SSVEPs is also possible. So in that sense, we are sort of covering both possibilities of high numbers of electrode in the lab versus small discrete setups that could be worn in everyday life. So what I'm presenting today is a first proof of concept using these LCD glasses. Here we just wanted to demonstrate the difference between walking and standing. So we had subjects walking and standing in a room with the glasses flickering at 10 Hertz. Participants were viewing the room with either the left or the right side of the room illuminated with an adjustable spotlight, and there was no other source of light in the room. So this was done for two reasons. Mainly, we wanted two visually distinct conditions to see if we could distinguish between them. But secondly, we also wanted to see if there were any differences when the left or the right visual hemifield was the dominant source of information. So the idea is that we can average enough of these trials, enough individual flickers of the 10 Hertz flicker, such that any movement artifact will be averaged to zero and be comparable to the SSVEPs while standing. The first thing we need to do is establish that this is the case. We need to know how many segments we need to average away this noise. So whenever we're faced with data corrupted by artifacts, the key question we should always ask is, how do we know the artifact has been removed? So when removing an artifact, there's always the possibility that there's either some residual noise in the data, which can be mistaken for neural activity, or that there's some neural activity which has been removed in the process of artifact removal. So here we used a signal generator. This is a device that delivers a small voltage across a scalp, which can be adjusted such that it gives a signal the same size as real EEG. Now this fake EEG will be the same when walking and standing, but the additional noise introduced by motion artifact will be added on top of the signal whilst walking. Now this signal is very small, it can't be seen in the raw EEG, but if we average enough segments, we can see this square wave signal on the top right. In this case, it's a square wave through a low-pass filter once we've averaged together enough segments. So we record this fake EEG whilst standing and average enough segments to get a clean signal, and this is what we call the true signal. And then we average more and more segments from the noisy EEG during walking, and eventually we will see the two waves converging and becoming the same. So here on the bottom right, the average difference between the two waves converges on zero as more and more trials are used, until eventually the average difference is less than a fraction of a microvolt, and this is then the number of trials we should use if we are generating the real SSVEPs so that we can trust the signal is not corrupted by motion artifacts. And just for context here, with 10 Hz flicker, 2000 segments is approximately 3.5 minutes. So we always aim for this high number of segments when we're collecting the real data. So once we've established how many trials we need with the simulated data, we can look at the real SSVEPs. So there's two ways in which we can analyze SSVEP results. One is a simple measure of amplitude, which is the equivalent of traditional power used in most EEG experiments. And the second is we can look at the waveform shape. So we can look at changes in the phase or the actual shape of the signal. Firstly, I'll just show example data from one individual on the top right. And this illustrates nicely the main finding. We found that the waveform shape and the phase was clearly different depending on whether the left or the right side of the room was illuminated, shown in blue and red. This means distinct signals can be derived from different visual scenes from the single electrode level. And secondly, this phase difference was largely preserved whilst walking. And this was true for almost every subject. At the group level, we found a significant reduction in SSVEP amplitude during walking, but only in right parietal electrodes. And also this was only significant during left hemifield illumination. That is to say, the primary source of visual information was the left visual hemifield corresponding to the right hemisphere. So we're seeing a clear lateralization effect. We want to know what's going on here. There's many reasons that could be contributing to these differences, but one particularly important one, and that we're interested in, is the input of the vestibular system. So this work was carried out at the German Center for Vertigo and Balance Disorders in the lab of Paul Taylor, and in this department we're interested in the vestibular system, that is the organs of the inner ear which sense movement through acceleration, such as the otoliths and semicircular canals. Now, information from the vestibular system is particularly important for navigation and orientation in space, but studies with naturalistic vestibular system are generally missing from the literature. So although treadmills and motion platforms are useful experimentally, they don't fully capture the natural vestibular input that we get when we're really walking, such as forward acceleration. Now, our hypothesis here is that the cortex is receiving input from the vestibular system when we're walking, and this input is absent when we're stationary. And like many cortical functions, vestibular processing is lateralized, with one hemisphere playing a dominant role. The right lateralization that we've seen is consistent with the literature on visual vestibular interaction, where right parietal regions in the cortex, in right-handers, are known to be preferentially process vestibular information. We were exclusively using right-handed participants here. And in previous research, we've shown lateralization in the alpha range due to the illusion of self-movement. So here we've replicated that effect with real movement. So conclusions of this study are, with LCD glasses, we can get SSVEPs of real-world scenes. These SSVEPs can be recovered whilst walking, and SSVEPs in the alpha range show a sensitivity to movement in the right parietal cortex in particular. And left parietal electrodes were significantly sensitive to differences in left and right hemifield illumination, again showing evidence of lateralization. So this was just a proof of principle. So what's next? The eventual goal is to work towards a system which can be worn in everyday life. So to do this, we need to demonstrate how many trials are needed to overcome the various types of artifacts that come with all types of eye and head movements, not just walking. Also, we need to track the variability of real-world scenes and investigate how much information can be extracted from this measure. And a long-term goal here is to make glasses which are a bit more like normal sunglasses, so people would be okay wearing them about their normal day, perhaps fitted over the top of normal eyeglasses. And then we can combine this with a small, discrete amplifier which can be worn throughout the day without disrupting normal daily activities. So eventually we would like to have something that we could put onto people that they could wear and go out, spend the day exploring an environment or doing what they would normally do, and collect artifact-free data. So this proof of concept used the alpha range because this is where SSVEPs are the largest, so it's a convenient starting point, but SSVEPs can also be generated in the gamma range. Now this is more difficult because gamma oscillations are much smaller than those in the alpha range. So here we can see SSVEPs from glasses flickering at 10 and 35 Hz just to show the difference in size. And gamma oscillations are particularly problematic for mobile EEG because muscle activity dominates in the gamma range. Again, SSVEPs can give the required signal-to-noise ratio. It's just a question of how many trials and getting enough trials. So the last slide I'd like to show is unpublished data that I'm still in the process of analyzing. And this is a pilot to demonstrate that mobile SSVEPs in the gamma range can be generated whilst walking around a real environment. In this case, the University Hospital here in Munich. It's a big building, we've got lots of long corridors, and we have people walking up and down, and we can successfully recover SSVEPs in the range between 30 and 55 Hz whilst walking and standing, controlling for visual input. Now, gamma range flicker has a further advantage in that above 40 Hz the flicker is not really perceptible. It's above the flicker fusion frequency, and it's perceived as a dimming of light, not a flicker, so it's far less distracting to be worn in everyday life. And gamma oscillations are particularly interesting because they're involved in a large number of cognitive functions, such as working memory and spatial attention. And of particular interest is that recent research has shown that 40 Hz flicker in mice can reduce the symptoms of Alzheimer's disease. Now this raises the possibility that as well as being experimentally interesting, there's a potential therapeutic application for LCD glasses to generate SSVEPs in the gamma range in people during their normal life, and this could be used perhaps as a diagnostic tool or maybe even something that has therapeutic benefit. So that's sort of a long-term research plan. There's many possibilities of what we can do with glasses once we can get rid of the artifact and have something truly mobile. So I'm always happy to hear from potential collaborators. If anyone is interested in using this method, please do get in touch. And thanks to Paul Taylor and the DFG and everyone at the Schwindel Ambulance. And thank you for your attention.